Okay, so we are going to walk through the kind of origins of the nucleus for the eukaryotes. Remember, the nucleus is what distinguishes the eukaryotes from the prokaryotes, is having that true nucleus. And so we'll have kind of a ring of genetic material if we're thinking about prokaryotes. And then the prokaryote also, we're going to have a membrane, a plasma membrane. Okay, so that's it. That's the prokaryote form. And what we think happened is simply kind of a process of invagination around the genetic material. So we have our genetic material, and then here's our plasma membrane, and we just think it started to invaginate, which isn't, that hard to believe, especially given that we see the more membrane that a prokaryote would have, the more surface area for, carrying on different metabolism, different reactions, so that they could kind of put all of the machinery together on their plasma membrane. And so these invaginations would occur around that nuclear envelope and would just become more and more pronounced, and would kind of start to then also expand around that envelope. And so again, we're just, dealing with all of those invaginations. And, we have some evidence that seems plausible given the structure of our current eukaryotic cell. And that is, when you look at the nucleus of a eukaryote, you see that there's actually pores around that nuclear envelope. And so in this drawing that I'm making here, those pores would, be the gaps where that membrane had folded in but didn't quite reach one another. The other part of this is that we then have an endomembrane system that is completely connected throughout the entire cell. And so if you think about the different organelles of the cell, right, we have that nucleus with its membrane bound center with the nuclear pores, and that membrane is continuous with the endoplasmic reticulum, and the origin of the vesicles that bud off from that endoplasmic reticulum is part of this membrane. And this same membrane will then fuse with the Golgi, or the Dictiosomes, we're talking about the tiny ones in plants. You may not have heard that word yet, so I'll write that out. Dictiosome are the tiny, tiny ones in plants, whereas in a lot of the animal cells you'll get Golgi bodies or a Golgi apparatus, which is a really, really large folding of membranes, where we have a lot of packaging and processing happening. And then we have that cell membrane, the plasma membrane on the outside. So everything in the cell, all of that membrane, is thought to be interconnected, be part of the same system. The folds in that membrane really help to kind of compartmentalize areas of the cell so that, for example, if you're thinking about compartmentalize, if you're thinking about digesting certain products, maybe food that you brought in if you're an animal or a heterotroph, then you don't want to digest everything else in your cell. So keeping things compartmentalized is a good idea. But basically this is the process that we think occurred leading up to the nucleus and the eukaryote and its endomembrane system.